Okay, so welcome to this next video in which we are discussing acyl-biotin exchange chemistry. Okay, so what we are trying to do is find out which proteins within a cell are palmitoylated. And the way we're going to do this is by exchanging the palmitoyl groups for a molecule which contains a biotin moiety and then detect the biotin moiety. Okay, the problem is that this molecule that we are going to exchange for the palmitoyl groups binds to any old cysteine residue. Okay, so the first thing we're going to have to do is neutralize all of the other cysteine residues within the proteins so that it's not going to bind to those. Okay, so here is my real structure of protein A. And now let me draw out, similarly, a real structure for protein B, so I'll try and keep them into their shape. So here's protein B, still in a triangle, and now I've got to try and draw a circle. Okay, I'll just do it like that. That's the best I can do, and here's protein C. So protein B and protein C have loads of free cysteine residues. So for instance, here might be a cysteine residue on protein B, here might be a cysteine residue on protein C. And also protein A will have loads of cysteine residues which are not palmitoylated. So we need to neutralize these cysteine residues. Now how are we going to do that? Well, basically we're going to bind a cysteine reactive reagent known as N-ethylmalamide onto these cysteine residues. So let me show you the structure of N-ethylmalamide, and then I'll show you how it binds to cysteine residues. Now, we'll start off with the structure of malamide first. Okay, and this is a really quite beautiful molecule. If you like chemistry, this will... if you like chemical structures, this will impress you. It's a beautiful, beautiful molecule. Okay, so, the structure of malamide, and we'll draw its skeletal formula because it looks prettiest when we draw its skeletal formula. So you have at the center of the malamide molecule a 5-membered ring, which consists of 1 nitrogen and 4 carbons. You then have a double bond at the base down here, and then off this carbon you then have a carbonyl group, and you also have a carbonyl group coming off this carbon here, like so. And then off this nitrogen you then have a hydrogen. Okay, so it's beautifully symmetric, this molecule. So this is the molecule of malamide. Now we're not going to use pure malamide. Instead, we're going to use what is known as N-ethylmalamide, which is often abbreviated to NEM for short. Okay, so this is N-ethylmalamide, or NEM for short. Okay, and the structure of it, well, it's going to be based on malamide, and basically what we're going to do is add on an ethyl group onto the nitrogen within this 5-membered ring. Okay, so I'll draw out this 5-membered ring again. Okay, we have a double bond there, and a carbonyl group here, and also a carbonyl group here as well. And then off the nitrogen you then have an ethyl group. So 1 carbon here, 1 carbon here. That's a 2-carbon molecule, that's an ethyl group. Okay, so here we have N-ethylmalamide. Okay, now we're going to mix this substance in with into our protein sample. Okay, so it's going to be mixed in with protein A, protein B, and protein C in our example, of course. But of course, when you're really doing this, you'll have huge, great protein samples which will contain far more than 3 proteins. But to get the concept across, we only need 3 proteins. Okay, so what's going to happen is this N-ethylmalamide is then going to bind to any free cysteine residues that are within the protein. So let me show you how this is going to happen. Okay, so let me start by drawing out a cysteine residue here, and I'll draw it on its side now. So here's the amino group, here's the alpha carbon, here's the carboxylic acid group, and now here is the R group off the side of the cysteine residue, which will be a methylene group with then a thiol group coming off it. Now, what you're going to do basically, and temporarily it's going to be helpful to sort of break with the skeletal structure a bit and put the hydrogens on here. Okay, So of course there were hydrogens coming off these carbons, but in the skeletal structure you don't have to show those. So I'm just breaking with the skeletal structure for a moment and putting those hydrogens there because it's going to help. Okay, so basically what we're going to do is, you can imagine breaking this bond between the sulfur and the hydrogen and sending one electron back to the sulfur and one back to the hydrogen. And of course, again, this is not an electronic mechanism. It's just to make sense of the reaction. Okay, then you can imagine breaking the second of these two bonds in this double bond here. So you're only breaking one, you're not breaking both bonds. Okay, so you break the second of the two bonds here, and you can imagine sending one electron back to this carbon and one back to this carbon. So both of these carbons now have 3 electrons. You're going to now bind this sulfur atom to one of these carbons. So it's completely symmetric, so it's arbitrary which one you choose, so we'll choose this one. Okay, so the sulfur atom is now going to bind to this carbon and will bind the hydrogen to the other one, so the hydrogen will then come and bind to the other one. So let's now draw out what you'll end up with. Okay, so again, here is the cysteine residue, so here's the amino group, here's the alpha carbon with its hydrogen coming off it, here's the carboxylic acid group linked to the next amino acid along, and now the R group of this cysteine residue, we have our methylene group, and now we have this sulfur atom which is now linked to this N-ethylmalamide molecule. Okay, so it will be linked now to this 5-membered ring, okay, which I'll draw skeletally again, okay, just because it's easier. So then here's the nitrogen, and we have carbonyl groups still here, so those haven't been affected. Okay, and we then have our ethyl group up here. Now, this double bond has gone, it's only a single bond now, so off this carbon you now have 2 hydrogens coming off, and off this carbon you have a sulfur atom and a single hydrogen. Okay, so that now is the structure that we have created. Okay, so when we mix the N-ethylmalamide with our proteins, okay, so let's draw our proteins out again. So we had protein A here, okay, and we had a free cysteine residue here, and we had 2 cysteine residues here which were palmitoylated. Okay, and of course you'll have far more cysteine residues than that in the protein, but to get the point across we only need 1 cysteine residue. And now what you're going to do is bind this N-ethylmalamide molecule onto that cysteine residue. And this N-ethylmalamide molecule will bind to all 3 cysteine molecules, but it will not it will not bind to the cysteine residues that are palmitoylated. So it will not displace the palmitoyl groups to bind to those cysteine residues. Okay, and this won't just happen on protein A, it will also happen on protein B. So if we've got protein B over here, and B has a free cysteine residue, then again, that free cysteine residue will get an N-ethylmalamide molecule bound to its bound to its phyl group. Okay, and again for protein C, all of the free cysteine residues in protein C will also get N-ethylmalamide molecules bound to their phyl groups. Okay, so that basically neutralizes the free cysteine residues. Now what we're going to do is we're going to split up our sample into 2 basically. So what we have done is we've homogenized our cells, we've got this huge great sample containing proteins now, and so far we've mixed in N-ethylmalamide to neutralize all of the free cysteine residues on these proteins. And now what we're going to do is simply break up the sample into 2. Okay, so even though I've just drawn the sample as 3 proteins, of course, in reality there'll be far more than just 3 types of protein, and also in our sample there'll be multiple of each type of protein. So for instance, in our sample there will actually be hundreds and thousands, if not gazillions, of A molecules. Okay, there'll be thousands, millions, billions probably of B molecules, etc. So it's not as though I'm going to, put the A and the B into one and then the C into the other. No, both of the samples that I'm producing, I'm just literally pouring our test tube into 2 test tubes now, and both of these new test tubes will get A, B, and C molecules in. So this will have loads of A, B, and C molecules in, and this will have loads of A, B, and C molecules in. Now, they're going to go down different routes now. Okay, so one is going to be our control, and you'll see later why we need a control. Okay, and the other, we're actually going to do something to it. Okay, so the control, we're going to do nothing in this next step. And the other one, what we're going to now add in, is a molecule which will break off the palmitoyl groups from the cysteine residues. Okay, and this is a molecule known as hydroxylamine. Okay, so let me show you the structure of hydroxylamine. Now, it's often written like so. H2N, which is an amino group, and then OH for an alcohol group, and that pretty much tells you its structure. I will draw out its structure just to make things crystal clear. So basically you have an amino group, like so, and then an alcohol group coming off the amino group, and this is the structure of hydroxylamine. Okay? Now, basically we are going to use hydroxylamine to break the palmitoyl groups off our cysteine residues. So let me draw here a cysteine residue with a palmitoyl group bound to it, and then I'll show you the way that this molecule is going to cleave the two apart. So, again, here is our amino group. Here is the alpha carbon with a hydrogen coming off it. And here is the carboxylic acid group bound to the next amino acid along. Okay, and the R group of our cysteine amino acid, you have a methylene group, and then the thiol group is linked via a thioester bond to the palmitic acid molecule. Okay, so there's the carboxylic acid group, and then you have 14 methylene groups, like so. So there's one, and I'll bracket it and put a 14 there, and then finally you have a methyl group on the end. Now, what you're now going to do is expose your proteins to hydroxylamine in this test tube, but not in this test tube. And what's going to happen is this molecule is going to break this palmitoyl group off these cysteine residues. So let me show you the reaction that's going to occur. Basically, you can imagine splitting this bond between the sulfur atom and the carbon atom. Okay? And imagine sending one of the electrons back to the sulfur and one back to the carbon. Then you're also going to break the bond between the nitrogen and the hydrogen. And again, you can imagine sending one of the electrons back to the nitrogen and one back to the hydrogen. Now what you're going to do is bind this hydrogen back onto the sulfur atom to create the thiol group back again. So let me draw this. Okay, so we're going to create our cysteine residue back again. Okay, so here is our alpha carbon, here's our carboxylic acid group, and here is our cysteine residue recreated. Okay, so basically it's just knocking the palmitoyl groups off the cysteine residues. Now what's the other product? Well, you're going to bind this nitrogen now to this carbon. Okay, so let me show this. I'm going to flip it round because it would be more normal to draw it the other way round. So here's the methyl group. Then we have our 14 methylene groups here. Okay, like so. And then you have the carboxylic acid group, which is now linked to that nitrogen atom, which has a hydrogen coming off it, and then an alcohol group coming off it like that. Now, this is an example of a molecule known as a hydroxamic acid. Okay, so this is called a hydroxamic acid. Okay, so whenever you have any R group there, this would be called a hydroxamic acid. So effectively it's an amide, but you've taken one of the hydrogens off and replaced it with an alcohol group. Now I want to stress that this name, hydroxamic acid, is not specific to this molecule. This is just the name of what this generic type of molecule is. You could replace this 15 carbon tail with any old R group and it would still be referred to as a hydroxamic acid. So this byproduct is a hydroxamic acid. Okay, now that's not very important. The important thing is that we have now re-exposed the cysteine residues where the palmitoyl groups were bound. So let me get another piece of paper and I'll show you what now has happened to protein A, protein B and protein C in this test tube into which we have added hydroxylamine. Okay, so protein A then, we'll start off with protein A. So here is protein A. Okay, now remember that the cysteine residues that were initially free have still got N-ethylamide bound to them, and the addition of the hydroxylamine does not affect the binding of N-ethylamide. Okay, that's very important. But now the palmitic acid molecules have been knocked off of these two cysteines up here. So remember, in my previous drawing, we had two palmitoyl groups attached here, but they've now been knocked off, and you've now got these two free cysteines again. Now, in protein B and protein C, you did not have any palmitoyl groups, okay? So all of your cysteine residues were initially free, so all of your cysteine residues still have N-ethylamide bound to them. So protein B does not have any free cysteine residues within it. And it's the same story for protein C, so over here, now let's have this as protein C. Again, all of the cysteine residues in protein C were initially free. None of them had palmitoyl groups bound to them, so they will have all got N-ethylamide bound to them, and the hydroxylamine does not knock the N-ethylamide off those cysteine residues. So now the only cysteine residues which are free within the proteins are those which originally had palmitoyl groups added onto them. Okay, we will now continue this discussion in the next video, where we'll see that we can now add on this molecule that contains biotin onto the cysteine residues which are now free, which are those that originally had palmitoyl groups added onto them.